continue going through the TCA cycle. So this brings us back to that first question about if we were to mutate just PFK, there's other places that help to regulate the process as well. So, there's no one point of regulation, there's sort of backups for the regulation. So then the last kind of topic with, when we talk about glycolysis, you can see glycolysis, TCA cycle, this entire cell respiration pathway is centered on glucose, right? It is carbohydrate metabolism. So what happens when people stop eating carbs? And I know there's lots of diets, right? We'll probably remember Atkins or South Beach diet. Now, anybody know the diet, the low carb diet that's very popular right now? Keto. So keto diet is another one. Keto is different from Atkins. In Atkins diet, they try to replace carbohydrates with protein, high protein levels. With keto, you're actually supposed to also keep your protein, you're replacing your carbs with fat. That's the goal of the keto diet. So what happens to all of this when you stop taking in so much carbohydrate? Well, one of the things that can happen, so this TCA cycle will be affected because oxaloacetate, which remember is the 4-carbon, that comes from pyruvate, which actually comes from glucose. So if you cut back on glucose, you will have decreased levels of pyruvate, which means you'll have decreased levels of oxaloacetate. So what can build up, if you look at that picture there, if you don't have oxaloacetate, which is a requirement for the Krebs cycle, what intermediate is going to build up in your body? Well, we can't get to citrate because we don't have the oxaloacetate, but what's before that? Acetyl-CoA. So you will have an increase in acetyl-CoA. So that's what goes in the blank here. Limited oxaloacetate means that acetyl-CoA will build up. And acetyl-CoA can actually go through this alternate pathway called ketogenesis, which is, probably sounds familiar from the keto diet. What happens in ketogenesis is that you start producing these other intermediates. You take your acetyl-CoA, which builds up, and you can produce acetoacetate. That can then be made into beta-hydroxybutyrate and acetone. Anybody know where you find acetone? In your fingernail polish removers, right? This is a byproduct of ketogenesis. And so one of the reasons, so people who are on low carb diets sometimes have, we call it like a ketone breath, or and it smells volatile. It's because some of the byproducts that you produce this is very, very low carb diets. You can produce acetone as a byproduct of this. Now people do, when they switch into this ketogenesis, they do lose weight because they start breaking down fats. Your fats are made of fatty acids, and fatty acids are broken down to acetyl-CoA. So it is effective in the short term for burning fat. There's a lot of questions though, long term, what are you doing? I mean, we know with fats, that certain fats are not healthy for us, right? So if you talk about saturated fats or trans fats, those are considered to be harmful. So I think if my word to the wise is if you're gonna do any of these diets, just to be aware of the types of fats that you are taking into your body. And I would, plant-based fats tend to be healthier than animal-based fats, like cheeses and meats. So anyway, it's kind of just the interesting. Anybody know people who do the keto diet? Yeah.
No, but I saw there was a study where they did a comparison of diets where there was just lower calories, and then there was a keto diet. They're about the same. They both level out, or the keto diet actually leveled out more than the just lower carb.
Yeah. I always find it interesting because I just feel that if you're going to have a new eating, it's just your lifestyle, you're going to change the way you eat, that's going to be sustainable. But for me personally, could I sustain a diet like this? I could not sustain it. So eventually I would kind of switch back to eating other foods. And then the question is, what happens with your metabolism when you switch back? Or are you going to gain weight back? And not that I mean, my goal is always health and, what are the healthiest foods to eat. But for people who are trying to lose weight, just considering all of the different repercussions for, you know, drastic changes. So anyway, so far this is the last slide that I have here. I kind of want to do the math and add up what we have on our chart. So at the end of the TCA cycle, all that we have is going to go into, it's either going to go into the electron transport chain, or, you know, it's the carbon that we've already gotten rid of. So at the bottom of these columns here, even though we haven't yet done ETC, if you want to add up how many CO2 have been produced total, and how many NADH total, and FADH2. So tally those up, because that's what's going to end up going into the electron transport chain.
Are they counting for the doubles or?
Yeah, so you're counting like from one molecule of glucose. So how many total CO2 do we have so far? 6. We've gotten, so that 6 that came in with glucose, they're all oxidized at this point. For NADH, how many do we have so far? 10 NADH that are, you can see those there as well. So the glycolysis, we got 2, pyruvate oxidation 2, and then we have a total of 6 from the TCA cycle. So that's 10. And then FADH2, we only have just the TCA cycles where that's produced. So we have 2 of those. So going into the ETC for the second question here, you actually could answer this second question. Each molecule of NADH can generate enough proton motive force, when we actually talk about building up that electron transport chain, that electrochemical gradient, I should say. So every NADH can give you 3 ATP, theoretically, and every FADH2 could theoretically give you enough energy to produce 2 ATP. So how many, based on the numbers you just calculated, how many total ATP should we be able to make in the electron transport chain? So you have to take your numbers 10 and 2, and then you're going to multiply by the numbers they have here. So 3 for every NADH, and then 2 for every FADH2. So what's that total number?
That's 36.
36, right? So that is, yeah, 3 times 10 is 30. And then 2 times 2 is 4, right? So 34. If we then add the ATP we got here, glycolysis, that's 2 more, and there's 2 more here. So that's where we get that number that you often see in a textbook that says 38. It comes from 34 theoretical that could be produced at ETC. These 4, and then we will talk, tomorrow we're going to talk about ETC, so we won't talk about it right now, but we'll talk about why we actually don't make quite that much. It's not perfectly efficient. Okay, so I think that's where we can end today. Just a note about this lab exam, because I realized I don't think I added the bonus points. The way that I calculated it was your individual score was out of 50, and I added those points to half the value that you got from taking it as a group. And then I checked to make sure that if your individual score was higher than what it would be if we add in the group score, then I would just take your individual score. But actually for everybody, it was either exactly the same or it was higher with adding in the group score. But I did not add your bonus points. So if you had pluses on the bottom 2 questions, you can bring it up to me and I'll add that on top of the score that you have. Because I guess I meant to do that and I never did before handing it back. So, yeah, it was more for the individual, yeah.
So there's a fresh.